Okay, so from here we want to have a look at a bit more detail the mechanisms by which insulin has its effect on the body. Okay, so we'll start with the basic fundamental processes of the breakdown and build-up of glucose, which is basically a sugar, carbohydrate substrate that we use to generate energy. So we consume food, essentially turn that into glucose, which circulates in our bloodstream and we use as an energy source. So we'll start with the terminology used to describe interactions with glucose and its breakdown and build-up. Okay, so first of all, we'll start with glucose and how it can be built up into glycogen as a storage substrate. Okay, so if we go with glucose being built up into glycogen, the terminology we use for that is glycogenesis, as in the generation of glycogen. Okay, for the reverse effect of glycogen being broken down into glucose molecules, you can see I've sort of depicted it a bit like a honeycomb lattice. We've got glycogen as just the stored form of multiple glucose molecules into a larger molecule. So the breakdown of glycogen into glucose is referred to as glycogenolysis, as in the lysis of glycogen. The next one we'll go to is when glucose is actually broken down into its own products, the end product being two pyruvate molecules, we refer to that as glycolysis. And then the last one we have, which is the build-up of products into glucose and is referred to as gluconeogenesis. Gluco being from glucose, neo being new, and genesis is in generation. Now, if we talk about the cells on which insulin actually has a function, we can start with the hepatocyte because insulin is secreted directly into the portal vein, so therefore its main impact is, or it is first at the liver, and that's where the majority of it has its impact. Okay, so in the liver we actually have a GLUT2 transporter, which is non-insulin dependent, so we have glucose moving into the cell via that, and we're going to draw this pathway in green. So there are basically 4 functions of insulin on a hepatocyte, and we're going to go through them as glucose moves into the cell. So we have glucose already moving into the cell, and one of the main functions of insulin is to promote the breakdown of glucose into its molecules, which is called glycolysis, and that is step 1 of insulin function in a hepatocyte. Now, normally in a hepatocyte as well, we've got the ability of glucose to be stored as glycogen and also the ability of glycogen to be broken down into glucose. Insulin, however, has a negative effect on the breakdown of glycogen into glucose. So we actually have an inhibitory effect here, which is step 2 of insulin function on the hepatocyte. Okay, step 3 is to do with amino acid formation into proteins, so protein synthesis, and we don't go into a whole lot of detail in terms of the mechanism of that, so we'll just summarise that very briefly there. Okay, so 1, 2, 3, and our fourth one is the basically fat synthesis within the liver. So that's via pyruvate. This is a much more complex pathway than I'm presenting here, but I'm just trying to give you the basic sort of fundamental idea. So we've got pyruvate normally go via a pathway, becomes a fatty acid, which then becomes a triglyceride, or fatty acids can enter to form energy, can enter the citric acid cycle. However, insulin has an inhibitory effect here, so we don't have fatty acids entering this citric acid cycle and producing ketones like they normally would. And I'm just mentioning this for future reference when we talk about diabetes. This becomes more important. So we're just going to note here that insulin is decreasing fatty acids usage into the citric acid cycle, and that's what we call beta oxidation in there. So we don't have the formation of these ketone bodies. Instead, it follows the pathway here to triglycerides, and triglycerides either form lipid droplets as storage, or they can enter the bloodstream as very low density lipoproteins. Very low density lipoproteins which can be transport these triglycerides around the bloodstream to other cells to use as substrate. So our four main functions of insulin in the hepatocyte, just to revise there, is glycolysis. We have one here. We have decreased glycogenolysis over here. That's 2. We have protein synthesis, amino acids, 3. And we have the formation of triglycerides and storage fat synthesis here, which is 4. Okay, now if we'd like to move on to muscle cell and the function of insulin in a muscle cell. So the muscle cell has a tyrosine kinase linked receptor for insulin, which we're going to talk about a bit more on the muscle cell in terms of its role rather than in the hepatocyte. The muscle cell is receptive to insulin, the molecule. So we'll just have the insulin coming in here. Beta unit, alpha unit insulin, disulfide bridging, disulfide, sorry. Now the step 1 of this pathway, so there is actually quite a more complex intracellular pathway that we're not going to go into in much detail in this series, just to simplify the general outcomes of insulin function on a muscle cell. Okay, so basically GLUT4, so the GLUT glucose transporters are expressed on vesicles within the muscle cell, and the activation of insulin on this receptor upregulates these transporters being carried to the cell surface membrane where they are expressed, and then glucose is able to enter the cell. And again, similar to the hepatocyte, we have increased glycolysis. And interestingly, instead of decreased glycogenolysis, we actually have increased glycogenesis in the muscle cell. Okay. Again, we do have the process of amino acids being converted to protein with protein synthesis, but we're not going to go into the complexity of that pathway in this series. Okay, so the summary of insulin function on our muscle cell basically boils down to the increased GLUT4 transporter expression on the cell surface membrane, so we'll call it step 1. We have increased glycolysis, and these aren't in any order, these are just to remember that there are 4 main functions of insulin on a muscle cell. We have this, we'll call that 3, and increased protein synthesis, so 4. So we have the 4 functions on the hepatocyte that we discussed previously, and here we have the 4 functions of insulin on a muscle cell. Okay, so we'll now go into the function of insulin in an adipocyte. Alright, so we have our adipocyte here, adipocyte, okay, and the first step again, in common with the muscle cell, is for the insulin to bind to the tyrosine kinase linked receptor over here. My drawings of insulin getting worse and worse. Okay, which promotes the transport of the GLUT4 receptor to the membrane of the cell and enables glucose influx into the cell. So that is the first mechanism insulin regulates on an adipocyte. The second one is increased glycolysis again. Now the pathway for that one is interesting. The rate limiting step, we're going to talk a little bit about the enzyme function here. Okay, so these 2 enzymes here are actually rate-limiting steps, just here and here, the phosphofructokinase and the pyruvate dehydrogenase. So what insulin actually does is upregulates this product here, fructose 2,6-biphosphate, which enables the increase in speed of glucose or glycolysis to pyruvate via these enzymes. So these enzymes are the rate-limiting step. So insulin upregulates fructose 2,6-biphosphate, which thereby upregulates these enzymes or increases their activity to increase glycolysis. So that is step 2. So now we're going to talk a little bit about the effect on fats in an adipocyte. So we have insulin directly increasing transport of lipoprotein lipase out of the adipocyte to be expressed on an endothelial cell. Now, if you remember earlier, we had very low density lipoproteins being circulated by the liver into the bloodstream. Well, now when they reach this adipocyte, they're actually able to be cleaved from those very low density lipoproteins into free fatty acids and glycerol. Okay, these free fatty acids are able to be transported intracellularly and contribute to the pathway of increased lipogenesis, so fat synthesis. So we get the formation of triglycerides within the cell and then into lipid droplets. And that is step 3 of insulin in the adipocyte. Step 4 actually comes via a negative regulatory mechanism of inhibition of hormone-sensitive lipase. So hormone-sensitive lipase's normal function is to cleave triglycerides into fatty acids. So what we have here is insulin actually decreasing this activity, therefore we maintain increased triglyceride formation. So that is the full function of insulin on an adipocyte. Okay, so just before we conclude, I'd like to just revisit the molecule here, fructose 2,6-biphosphate, and its influence on glucose breakdown. I'll just include the enzymes in grey that are involved. So for glycolysis, we've mentioned these two enzymes, phosphofructokinase here and pyruvate dehydrogenase. Two of the enzymes responsible for part of the pathway of glycolysis. So F26BP has positive effect on these enzymes, and on the FDPase on the other side, which is actually fructose 1,6-biphosphate. This is the same. That's the same as this FDPase enzyme here, which is responsible for part of the pathway of gluconeogenesis. We have this product having an inhibitory effect on that enzyme. So when we are producing more of this product, we actually get an increase in glycolysis. So as you might imagine, insulin upregulate the production of fructose 2,6-biphosphate and thereby have these effects. So if we revisit the general function of insulin, just to revise, it's all about decreasing blood plasma levels of glucose and amino acids and fatty acids, free fatty acids, and sequestering them into tissues that we can utilise them as energy. And that concludes the insulin function on our main cell receptors. So we have, we've discussed the hepatocyte reaction to insulin in the 4 steps. We've done the muscle cell 4 steps, and that was concludes the adipocyte 4 steps of insulin, just to try and simplify it there. And that concludes this episode for the series of insulin function.